Hi, today we will be discussing Chapter 28 of Harrison's Principles of Internal Medicine. This chapter deals with disorders of the eye. For this slide set, we will be discussing first the human visual system. The act of seeing begins with capture of images focused by the cornea and lens on a light-sensitive membrane in the back of the eye called the retina. Light is absorbed by pigment in two types of photoreceptors, the rods and the cones. In the human eye, there are 100 million rods and 5 million cones. The rods operate in dim or scotopic illumination, while the cones function under daylight or phototopic conditions. The cone system is specialized for color perception and high spatial resolution. The majority of these cones are within the macula, the portion of the retina that serves the central 10 degrees of vision. In the middle of the macula, a small pit termed fovea, packed exclusively with cones, provides the best visual acuity. Photoreceptors hyperpolarize in response to light, activating bipolar, amacrine, and horizontal cells in the inner nuclear layer. After processing, the flow of sensory information ultimately converges on a final common pathway, the ganglion cells. These cells translate the visual image impinging on the retina into a continuously varying barrage of action potentials that propagate along the primary optic pathway to visual centers within the brain. Ganglion cell axons sweep along the inner surface of the retina in the nerve fiber layer, exit at the optic disc, and travel through the optic nerve, optic chiasm, and optic tract to reach the targets in the brain. The majority of fibers synapse on cells in the lateral geniculate body. Cells in the lateral geniculate body project in turn to the primary visual cortex. Although the lateral geniculate body is the main target of the retina, separate classes of ganglion cells project to other subcortical visual nuclei in different functions. Ganglion cells that mediate pupillary constriction and circadian rhythms are light sensitive owing to a novel visual pigment, melanopsin. Pupil responses are mediated by input to the pretectal olivary nuclei in the midbrain. The pretectal nuclei send their output to Edinger-Westphal nuclei, which in turn provide parasympathetic innervation to the iris sphincter via an interneuron in the ciliary ganglion. Circadian rhythms are timed by a retinal projection to the suprachiasmatic nucleus. Visual orientation and eye movements are served by retinal input to the superior colliculi. Gaze stabilization and optokinetic reflexes are governed by a group of small retinal targets known collectively as the brainstem accessory optic system. The eyes must be rotated constantly within their orbits to place and maintain targets of visual interest on the fovea. This activity is called foveation or looking and is governed by an elaborate efferent motor system. Each eye is moved by 6 extraocular muscles that are supplied by cranial nerves from the oculomotor, trochlear, and abducens nuclei. Activity in this ocular motor nuclei is coordinated by pontine and midbrain mechanisms for smooth pursuit, saccades, and gaze stabilization during head and body movements. Large regions of the frontal and parieto-occipital cortex control these brainstem eye movement centers by providing descending supranuclear input. This is the end of this set. Next set for this series is clinical assessment and visual function.